if you take a look at these pictures, like the Recite book, the Nature publication, which is basically a scientific publication, for example, American Journal of Physics and Physiology, or the Science journal, and the thesis, the title said, what is the common between all these three? First of all, nothing, because when you are in the kitchen, it is fun, even if you're drinking a wine while you are just preparing your lunch. If you need to write a scientific publication, that's like, how can I say, mental breakdowns, nerve-wracking, and all the other sad stories behind. And the thesis work, it is almost the same thing like writing a scientific publication, but in like a rookie mode, I would say. And obviously, you really need to post this picture, otherwise it is not going to be valid, right? That you were suffering from years just to make this thesis work. But let's go back to the business and the serious part. What is common between that the research book, the publication, and even the thesis contains information as a kind of protocol, which whatever you do, whenever you do, wherever you do, it should give you the same result, theoretically, obviously. Because if you did a recipe, like a recipe just to make a muffin, the ingredients, the steps should be the same thing. And if somebody on the other part of the world is trying to make the same cake, then he or she needs to do the same thing at the end, right? So that goes for the science publication and also for the thesis works as well. As a teacher, I saw many times that somehow this is not the most sharp and professional part of these works, because some steps are missing or it is not really well written. And that is a problem. And that's why you are here and now you're watching this video. So let's go further and let's see what we can do. So if we were talking about the work in the kitchen and the work in the lab, it is the same thing. Just to mention that whatever you saw in those kind of movies and series, this is not going to happen in real life. So if in one day you can do the DNA analysis from an unknown sample without any problem, no, that's magic and that's not valid. But the magic and the valid part here comes on the same road, because whatever you do in the kitchen step by step, whatever you do in the lab step by step, it is the same thing. This is the protocol. So now let's take a look. This is what you're just writing in the lab, right? And now the reason why the shopping list on this screen, because whenever you do something, you need to use a kit, you need to use an antibody, you need to use a solution, you need to use whatever. So it is basically a shopping list. And nowadays, the most professional journals, they're asking you the so-called STAR method, in which you really need to list all those antibodies, kits, solutions, liquids, whatever you used, with the source and with the catalog number, just to make sure that, for example, another researcher or somewhere else, they can reproduce your data. So this is the new aspect of research and education, but that should go on the same way for the TDK work, the thesis work, the PhD dissertation as well. So there are some formal requirements, for example, this protocol and the precise professional protocols that you need to write it in. But as you see, we have different aims, because TDK work, thesis work, they are like a shorter version, I would say. Manuscript, which is the scientific publication, this is a different level of the writing aspect. PhD dissertation or the MTA doctor, this isn't like the higher level, but both, like the small one, the intermediate, and the high level of these kind of scientific works, they have almost the same formal requirements. You really need to have an abstract, then you need to have an introduction, material and methods, results. You need to conclude your results, and obviously you need to have the acknowledgement and the author contribution and the references. But the reason why there are two acknowledgements here, because usually it's not just like you're just going to acknowledge that, oh yeah, I'm so grateful my mom that she raised me and she told me that I will be a good researcher. No, this is not the way like that. You need to acknowledge all those colleagues who helped you to put the manuscript together, or the ones who were correcting it and reviewing it. So there are like different levels of this one. But most of the cases, these type of formal requirements are there. What you really need to know in the case of the TDK and the thesis work, that besides this one, you really need to have an appendix in which you're clarifying that, hey, I was doing the one, I mean, I was the one who is doing this in the lab. I was there in the night when I was doing this. I've made the figures and everything. In the case of the PhD dissertation and the MTA doctoral work, the scheme is the same, but it is not that strict anymore. The reason behind is, I think it's easy to understand, because when you have a PhD dissertation, it means that you already did some manuscripts together. So in that sense, it is not that strict. I would say that the most strict is the manuscript and the thesis work. But there is one thing which is really common, the material and the methods. That should be written as professional as you can do. Otherwise, they're going to criticize it. And this is basically the result. So, but how are you going to do this? Because, I was a student too. And what happened, like, okay, I would have like 2 weeks to write it. So I'm going to do this for the whole day. Yeah, and then I'm going to wake up early, and then I'm going to start to read, and then I'm going to start to write, and then I'm going to create a folder, and then I'm going to write the storyline. I'm going to make the list of the figures, which is so theoretical and ideal, and then I'm going to make my final steps on the figure. Then I'm going to write the results. And if I'm done with everything, then I'm going to write the introduction, and let's talk about the discussion afterwards. And then I'm going to put together the abstract, and then I'm going to write the title. So this is not going to happen in real life. Be honest, you don't have time for that. You are going to be late, and you are going to make a rush at the really, really end. So in that case, if you take a look at this figure, which means like you can draw this cute little guy here, step by step by step by step, this is just an imagination. If you are not doing this like one by one, day by day, week by week, at the very last days, this whole list is going to be distracted. So if you take a look at this order, I would say that this is happening more in real life, that first somebody is giving you a title, and then you're going to put together the abstract, and then you're going to write the introduction part, and then the storyline, and then the results, discussion, list of figure, finalizing the figure, and create a folder. That happened with me as well. Like, I was not taking care of the organization part, so I did not organize my stuff, and I just put everything together, but there was no folder, so there was no logical order in these cases, and that made my life harder and more sad, and at the end I was so devastated. But if you do a logical order, then you can do some beneficial helping parts for yourself. So in real life, what is happening here, that if you join to how can I say, into a lab or like a research group, and you do some experiments, obviously what is happening, that they're going to check how these results are going to fit into any other research aspect, what they did before or they plan to do. And then regarding to that one, you are going to do the experiments, and then afterwards you are going to see like, okay, let's see, can we use this one, can we use that one? And if you can use something, then you are starting to put them together with the other preliminary data and the colleagues' work. And then you are going to target out what you want to convince, I mean, about like emphasize and convince the others, like, hey, believe me, this protein is going to do that, this protein is going to do that. But that's the sad truth in the case of the research. And then you're going to write the abstract, and then the introduction, and then you will finalize the figures. So There are different aspects of this whole research. It depends on the supervisor, it depends on the lab, but I'm pretty sure that all the educational system, they are just referring to the same thing, that you join somewhere. So based on the preliminary data, everything is already set up for you. Nowadays, there is a new way of writing all these things, and this is the Chat GPT. Maybe you know, maybe not, but most of the educational system and universities are struggling with the fact that people starting to use ChatGPT to write a scientific research paper and the thesis work. However, there is some regulation in which the scientific community regulates that who can be an author, and if somebody is using ChatGPT to write a scientific research paper, that should be categorized in a totally different way. But it is not going to uh, go with the thesis work. So how it goes? You register for the ChatGPT, and then he's going to, or she's going to, it depends on you how you consider the gender of the ChatGPT. And then how can I help you today? That is the first question. And then you start. For example, how to write a scientific paper. So after you click on enter, then it is going to give you like a full list that how you should do that. Choose a topic, review the literature, define the research question, design the study, collect and analyze data, organize the results, interpret the results, blah, 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 blah. I'm pretty sure that all these 17 steps, you are going to focusing on 4 or 5, and that's it. And then the supervisor is going to do the rest, or the supervisor is going to make your life more hard regarding to the changes what he or she wanted to do. But for example, if you're asking like a single thing, like what I did, what do we know about the retinoic acid receptors? First of all, it was a bit confusion here, like the chat GPT doesn't know like which rare you were asking for. And then as you see, based on the text, if you're referring to the receptor associated with the retinoic acid, then it is going in this way. So there is one thing which is good here, that ChatGPT is using all the information which is available online. But there is one thing. However, it looks okay. However, it looks valid, but it does not mean that the source of information is valid. Sometimes ChatGPT generates information which can be true, but the problem is it is not a referable source. So it is making the source, which does not exist. So please do check whether you write something. This is really something that you wrote it in an article or not. And now let's take a look for the first part, which is basically, I would say, this is the key of all the interpretation. So if you are in the Hungarian system, I'm pretty sure that if you are preparing for yourself for a language exam, that you had this picture describing method and task, which was really, for me, especially, I just don't see the point. They give me a picture and I need to describe like what is there. So in this picture, I would say, okay, that's a field and that's a boy and that's a girl. And maybe she's really young. I don't know how many years old, but that's it. Maybe she's playing. And afterwards, we need to generate a story behind on these language exams. But in research, it is a bit different. So first of all, you can like say what you will see on the figure. As I said, girl, ball, field, green, blue, blonde, blah, blah. And then playing, weird face, outdoor activity, parenting, physical education. But if you have a figure, the figure needs to talk for itself. And the figure legend, which is written underneath, it should describe everything which is relating to the picture and helps you to understand what is going on the picture. So now let's say what else you need to do if you have a figure. First of all, you need to have a title. And then what is represented on the figure? This is really crucial because people needs to know what is on the figure. However, it is obvious, you really need to write it down. Also, because these figures needs to be really easy to understand, like, I mean, super clear. you need to write it down how it was carried out and how many times it was carried out. Because if you give these two informations, people don't need to go backwards and backwards to the thesis work, like, okay, oh, well, what was this? What was this? And believe me, as a reviewer, this is the most annoying thing, because when you are reading a thesis work and they just didn't put anything underneath the figure, you really need to just, scroll back and like, what the heck is going on here? And besides this one, the significance and the test which was used for the significance, this is really crucial, because these are the major important parts to understand the figure and to trust the figure. So now let's see. Title. The title can be brief, it can be long, but usually it should be a brief title which applies to the entire figure, including all the panels. So, for example, if I give a title to this picture like, oh, the playing girls on the field and the boys, there is no boys on the picture, and also, like, this is really misleading. The title can be either descriptive or declarative. And basically, these two are totally different. What you see in the figure in the text, like the flow cytometry analysis or the viral protein usually decreases, blah, blah, blah. So the descriptive and the declarative has a two different aim when you are using that, but both are working really well. So now let's take a look how it can be. First of all, if you just go with the really easy way, like the blondie and the balls, early play with the balls helps the body posture. And then, to be honest, when I just made this presentation, I'm now concerning that maybe this was not the best picture I choose, because these titles are really, really weird. But if you go further, you can say like blondie playing with the balls. This is also really weird. Correct way of touching the ball. Again, really weird. So you really need to be clear, and you need to make like gentle but a general title. For example, correct body posture to keep during squats, or the correct execution of squat helps correcting the body posture. Now, this is how these titles are made. What is represented on the figure? If your figure is really complicated, then you really need to write something else, because the title won't be enough. And this is really, really important. A brief text can summarize what is visible on the figure. For example, here, the correct execution of a squat helps correcting the body posture executed by a 5-year-old female. For example, this one. If you go further, how it was carried out, this question targets the material and the methods. So if you describe which technique was used, and the description includes everything, what are the cell types, the animal models, the conditions, the treatments, how long it was treated, how long it was carried out, and so on, and all the numbers of the replicates, these are the most critical information, and these are helping you to understand the figure more. There are some exemptions. For example, in certain cases, the journal is asking you that, hey, can you just write the figure legends, but please do not include the method part, because that should go into the method section. But it depends on the journal that you are choosing. How many times it was carried out, or the signs of the significance, or the test used for the significance. Without this one, nobody is going to believe you what happens there, which means that you really need to do there, like how many repetition was done, it is a biological parallel or not, and what are the error bars. I think this is the weakest point of all the thesis work I read by so far, because people are not clarifying that this is the standard deviation, this is the standard error, so this is all what. Because if you are working with the standard deviation, it's not the same like the standard error and all the others. So please do take care of like which values are represented on the figure. And also the test which was used, like, okay, I was using the ANOVA. Which one? One way, two way. It is a multiple comparison or a single one. You use the t-test. Which t-test? The paired or the non-paired? The parametric, the non-parametric. And in that sense, if you're using variance analysis like the ANOVAs, you really need to go further, like, oh, yeah, I used something else. Maybe this is a post hoc test, for example, Bonferroni or the Tukey one. You need to write all these things. So now let's take a look at the scientific example. And because I don't want to choose all the others, so I choose one of our previous one. So on the part of the upper panel, you see that is a figure. As you see, many colors are there, many different things. And we really need to write down everything. So if you take a look at this is the figure legend down, and with the different color, I'm trying to represent the different parts of the figure legend that you really need to take care of. So here in bold, you see that the title is way more general. It says like the tissue transglutaminase expression moderates arsenic-induced apoptosis. I didn't say anything. I just said like, okay, the protein which I'm looking for, it has some effect on the apoptosis and even moderates. I did not say that it is enhancing, it is decreasing or attenuating. It is just moderates. So in the A part, where I was using like tissue sections and like these are like Giemsa-Grünwald staining slides, I just wrote down like these are representative images of May Grunwald Giemsa staining for morphological examination of these cells treated with this and this for 3 and 5 days. And I put how many times I repeated this one. Also, I target that the light microscopic images and the documentation were obtained using the Floyd Cell Imaging Station instrument and with the company. So it's really, really important to make sure that all the information is there just to understand that how this figure is made. And I think this is because people get super comfy about like, okay, I'm going to read an article, but they don't have time. So they are going to look at your figures and they need the information without reading all the other part of the paper that what is happening on that figure. So cell death features are more with different triangles. These are the triangles, right? On the color code listed in the lower panel. And the color code are here. So basically, I helped the reader, I hope that I helped the reader to find that which color shows which type of morphological changes. Also, quantification of May Grunwald Giemsa state cytopin slides. This is B1 and B2. From each side to spin slides, 200 cells were treated with this until that long in the combination, blah, blah, blah. So I already wrote down like, hey, that was the first part. And then I counted the cells and this is the result. But the graphs represent the mean values of the counted cells where the black, gray, orange colors mark the cell death features. So again, you need to target everything, which is a color there, which is like a shape there, because you really need to give all the information to the audience. And also, I just thought like vehicle controls are in the supplement figure. So if you want to read all the other parts, then go ahead, you will find it. And because I showed some significance here, sorry, then I need to type like, hey, statistical significance was determined by two-way analysis of variance and with this Bonferroni post hoc test. And because the numbers of the asterisks are really like sharp in that sense, I really need to make clear that what is the comparison between like what did I do? So I put here that this treatment was compared with this one. And this treatment was compared with this one. So basically these signs are showing the comparison between these two. So this is how a figure should have been done every single time. Obviously, if you have just a picture, that's a different story, but that will be in a later topic in the course. But if you have like a clear data with a graph, this is the one that you really need to take care of.